Hello everyone. Today we are going to discuss about the edible vaccine. It has been rightly said that one day child may get immunized by munching on foods instead of enduring shots. In simple language we can say by eating a simple food we will be getting immunized. So the concept came from when there was a concept about the edible vaccines. So, we can talk about the edible vaccines today. Edible vaccines are the components or subunit vaccines. What we do, we encode the genes of bacteria or viral in the plants, either in the fruits or in the vegetables, by which we are able to have edible vaccines. As with the development of genetic engineering in plants, we have developed, as such, we also know that we have developed many resistant plants. These plants, recombinant plants, have shown resistant towards the herbicide, insecticide, drought, frost, and many more things. So, by developing these edible vaccines, it will be orally taken at any time and inexpensive. This vaccine, edible vaccine, will be inexpensive vaccine for the developing world. So, by having edible vaccines, we will be able to cover large portion of the world which will not be having or which will not afford the vaccines. Due to this edible vaccines, we will be able to immunize those people as well. The peculiarity of this edible vaccine is like that it doesn't need refrigeration. They are heat stable. It has been estimated that every year there are death due to these infectious diseases is estimated about 17 million people. 17 million people die about of infectious diseases, in which 7 million are only children's. We have earlier we was using the cell culture or the recombinant vaccines, but they were too costly and they were we was not sure that whether they will get be attacked by the pathogen or like that, so that were not feasible as much as its edible vaccine is feasible. So important is that these edible vaccines are normally we eat food, so edible vaccines are used as food, but we are also getting immunized. This edible vaccine will successfully improve the quality of life. The expression of subunit antigen of infectious organism. We can take subunit gene of infectious organism. We will place this gene into the transgenic into the plant, which will be able to grow. So when the a plant will be able to grow a gene which is not its own, so these plants are called as the transgene. So these transgene, they will be able to produce the vaccines. These vaccines are having ability to initiate the immune response, in humans. So the first report about the edible vaccine, the first report about the edible vaccine. So in that case, what they have done, they have the first report everywhere in the plant tissue culture technology or in the plants. They have scientists have always used tobacco plant. So first report for edible vaccine was also from the tobacco plant. In tobacco plant, they have inserted a gene from Streptococcus mutans. They have used a gene from Streptococcus mutans. So even when this product was launched, they patented this under the International Patent Cooperation Treaty. So what we have scientists have taken the tobacco plant, in which they have transferred gene from Streptococcus mutans via the Agro bacterium mediated transformation. So when tobacco plant is having this it has been observed that they were able to initiate the immunological response, which were stimulated the production of surface antigen in the saliva. So this was how edible vaccines were introduced into the plants, or how we were able to have edible vaccines from the plants. Now, if we talk about the vaccine, we simply say it's a substance, any substance, which will be able to stimulate immune response when this substance is introduced in our body. These vaccines earlier we used to have injected vaccines or the oral vaccines, due to which they boost our immune system by generating antibodies against that specific organism which is responsible for that particular disease. If we will go into the history, 200 years back, we all know that Sir Edward Jenner in 1796, he was the one who discovered the principle of vaccine, and what he did during that time in 1796, there was a disease in cattle, cowpox. So even in that particular area, they were having a human disease that was called as smallpox, and it was extremely virulent disease with the high mortality rate. So he used cow powder cells and injected it to 8-year-old boy James Phillips. Later on, he observed that the boy was fully protected against the smallpox. So this is how Edward Jenner discovered the principle of vaccination. If we will go about the word vaccination, it's a Latin word. Veta means cow. It has been originated from the cow, so they called it as vaccination. Even it's not necessary. We can use even dead organism or any part of any organism that should have response to the immune system, that should be able to response to the immune system. So if we will talk about the edible vaccine, the first test of edible vaccine on human trial was carried in 1997, where scientists individually came or voluntarily came to have anti-diarrheal transgenic potato. So scientists first made at a human trial, they made a transgenic potato in which they inserted an anti-diarrheal gene. This product was launched by the Bovisi Thomas Institute at Cornell University. After consuming the potato, obviously antigens went into their body. They were able to stimulate or accelerate the immune system or increase the immunity in that particular person. It was simply as it revealed, or it received simply as a traditional vaccination process. So it simply gave the traditional vaccination process. Then it come into idea that we should be able to have edible vaccines. Charles was a scientist from the Texas A&M and University. He was the one who insisted, or who appealed to the World Health Organization, that we should be having vaccines, so that we should be having edible vaccines, so that the child of the developing countries will not suffer. So since then, the concept of edible vaccine came into the existence. So if we'll talk about the types of vaccines, we are having 4 different types of vaccines: live attenuated vaccines, or killed or inactivated vaccines, toxoid vaccine, and component vaccines. So if we will talk about the live attenuated vaccines, these are simply created from the naturally occurring organism. Even these attenuated vaccines still have ability to infect the people. What we do, we take a virus, We attenuate it or weakened, इसको कमज़ोर बनाता है by growing it again and again under laboratory conditions. We take organism virus which is virulent, we grow this virus in the lab conditions, मतलब under animal cell line culture में हम लोग इसको यूज़ करते हैं, और इधर plants mostly we do it in the animal cell or plant cell. Again and again इसको क्या करते हैं, we allow it to grow. So जब इसको ग्रोथ करते हैं इसकी विरुलेंस जो है वो कम हो जाती है, मतलब चेंज हो जाती है. वी कॉल दिस एज एटेन्यूएटेड सेल कल्चर. वी कॉल दिस एट जो हम लोग इसमें मीडिया यूज़ करते हैं, न्यूट्रिशन मीडिया यूज़ करते हैं, वी कॉल इट एटेन्यूएटेड सेल कल्चर. सो बाय दिस वी वर हैविंग organism that is less virulent, but that organism is having ability to initiate immune response in our body. So we are having different examples like measles vaccines, mumps vaccines, rubella, or we call this as German measles vaccines, oral polio vaccines, or we have varicella. This is name for chickenpox vaccine. So next is our killed or inactivated vaccine. Simply what we do, we simply take virus, inactivate it, kill it, मतलब सीधे organism को kill करता है. We can use chemicals like formaldehyde. Organism लिया उसको सीधे kill कर दिया, that then inserted in our body, that also act as a vaccine because the killed organism are having ability to generate, to accelerate the immune system. So we are having examples: inactivated polio vaccine. It is a short form of polio vaccine. Or we are having another example, inactivated influenza vaccine. Now the third class of vaccines are the toxoid vaccines. As the name indicates, what we do, toxoid means toxins which are produced by these organisms. We take these toxins, we simply heat it or we give the chemical treatment with the help of formaldehyde, so that this toxide will not be having ability to cause illness. But this toxide, when introduced in our body, will act as an antigen. So whenever we are having antigen, they will be able to initiate the immune response as the natural toxide will do. So by this way also we will be getting immunized. So we are having very common, we are having like examples, diphtheria toxoid vaccine, or we are having another class is the tetanus toxoid vaccine. So if we'll talk about component vaccine, as name indicates, component means any part of the virus or the bacteria. They can't, the part when we are using any component, these component vaccines are not able to cause any disease, but they are able to stimulate the immune response in our body. So we are having many examples: Haemophilus influenza type B vaccine, hepatitis B vaccine, hepatitis A vaccine, pneumococcal conjugate vaccines. We are having many different successful vaccines also. For vaccines like we are having vaccine for German measles, diphtheria, whooping cough, or tetanus, smallpox. But still we are having limitations. We are having many vaccines, but still we are having some limitations. Like all the organisms, infectious organism, we won't be able to grow these under the laboratory conditions. So if we are growing these vaccines, bacteria or viruses in the laboratory conditions, for those animal or human viruses, we need live cell cultures. We need live cell cultures, means see the living cell चाहिए, but that are too expensive. If we will go for the yield of these live cell cultures, yield is very low, and if yield is low, it means the production cost will be the very high. Finally the vaccine will be very costly, so it won't be available for the developing world people. So it's not even extremely safe as well as this live cell line may get exposed to pathogenic agents, due to which we will get altered vaccines or altered antigens vaccines which we are using. So sometimes it happens also when we are killing, there will be inefficient killing or inefficient attenuation of the vaccines. So we will be having in dilemma that the vaccine which are using is attenuated, but still that will be in a virulent form. Simply हमने क्या किया इसमें, what we do, हम लोग वायरल, हम लोग attenuate करते हैं ना या किल करते हैं. समटाइम्स ऐसा भी होता है कि सॉरी एटेन्यूएशन पूरी नहीं हुई होती है, उसकी वायलेंस वहीं पे होती है और गलती से हम लोग वो ऑर्गेनिज्म एज अ वैक्सीन इंट्रोड्यूस करते हैं इंसान की बॉडी में, तो सिंपली वो डिजीज़ क्रिएट कॉज करेगा, फिर इस वजह से डेथ भी हो सकती है. सो इट मीन्स वी आर हैविंग डिफरेंट लिमिटेशंस. इवन जो उसकी ये है, जो ये वैक्सीन हम लोग करते हैं, इसकी लिमिटेड शेल्फ लाइफ है. इवन हम लोग हर एक डिजीज़ को ट्रेडिशनल वैक्सीन से प्रिवेंट नहीं कर सकते. एज इन केस ऑफ एड्स, we can't prevent all diseases like AIDS through the traditional vaccine system. So these were some limitations about the traditional vaccinations. So scientists have then used the recombinant DNA technology into the plants, by which what we do, we are having many applications of this recombinant DNA technology. What we do, we take a virulence जीन एक लिया जिसका वायरुलेंस जीन है, उसका वो हिस्सा हटा दिया, कट कर दिया उस जीन का जो वायरुलेंस क्रिएट करता है. फिर आधा जो जीन है उसको प्लांट्स में डालेंगे, प्लांट्स उसके एंटीजन्स बनाएगा, लेकिन वो वायरल एंटीजन नहीं. देन दैट प्लांट पार्ट विल, और फॉर एग्जांपल वी हैव इंट्रोड्यूस दिस इन द बनाना. फिर बनाना हम लोग खाएंगे, उसकी वजह से क्या होगा, एंटीजन्स हमारे बॉडी में आएंगे, जिसकी वजह से फिर हमारी बॉडी क्या करेगी, इम्यून रिस्पॉन्स देगी. तो फिर इट मीन्स दिस विल बूस्ट आवर इम्यूनिटी एंड दे विल ऑल्सो बी एबल टू प्रोड्यूस द मेमोरी सेल्स. या हम लोग एक ऐसा सिस्टम बनाएंगे, एक ऐसा आर्टिफिशियल जीन बनाएंगे जो सिर्फ एंटीजन एज एक्ट करेगा, एंटीजन विद द हेल्प ऑफ पर्टिकुलर नॉन पैथोजेनिक कैरियर सिस्टम. तो फिर उसको इंट्रोड्यूस करेंगे, तो उसकी वजह से भी हमें क्या होगा, हमारी इम्यूनिटी बूस्ट हो जाएगी. नाउ इफ वी टॉक अबाउट द सब यूनिट वैक्सीन, सिंपली हमने हीट किल लिया और एटेन्यूएटेड वैक्सीन लिया, एटेन्यूएटेड ऑर्गेनिज्म लिया. सो दीज एटेन्यूएटेड और हीट किल्ड ऑर्गेनिज्म आर एबल टू इनिशिएट इम्यून रिस्पॉन्स. So we can say, in other words, we can say for vaccination, we don't need whole organism. We can use part of any organism. Like in case of vaccines, we can use as a subunit vaccine, we can use outer surface viral proteins. We can use outer surface viral or either capsid envelope proteins. These can be these can be used as a subunit vaccine. Like in diagram we are having, I have shown, we have having different nucleic acid. Without nucleic acid, we are having capsid proteins, will be the envelope proteins, will be different envelope proteins are there. These can be used as a subunit vaccines. Again, for subunit vaccines, we are having advantages as well as the disadvantages. So when we use subunit vaccine in a purified form, obviously it will be stable and safe. These subunit vaccines are well chemically defined. These are the free of exogenous proteins. Like in those subunit proteins, we don't have the nucleic acid. So when there is no nucleic acid, then this vaccine will not be able to initiate any virulence in humans. So if we will see the negative part of the subunit vaccine, so we have to go for the purification process. The purification of those subunit vaccines will be very costly. Whenever we are going for the purification, it means we have to use some chemicals, we have to go for downstream processing of those proteins. But it has sometimes it will be that when we are extracting these protein, there will be some conformational change. So it means that their antigen stays altered, then if antigen T is altered, that can't be used as a vaccine for a specific organism. So the concept then came from the edible vaccines. The edible vaccines are having many advantages. These edible vaccines are used with the help of recombinant DNA technology and with the help of subunit vaccines. We are able to grow edible vaccines in vegetable and fruits. Vegetable and what we do whenever there is infectious agent. So the first, whenever infectious agent is entering in our body, the first defense mechanism is our skin, or if it is into our the gut, then when it is inserted, the organism is getting in our body, it will be obviously through the respiratory tract. Mucous membrane of respiratory tract is there, or gastrointestinal tract or urinary tract. So these are the surfaces from which we are able to having entry of the organisms. Like here we are having organism. So if this organism entering in our body, we are having some specific mechanism that is already present in our body. The cells there are able to generate secretory cells able to generate secretory cells. We are having, for initially we are having, when we are having memory cells. These memory cell helps T cell to produce cytotoxic T cells to attack infected cells. हमारी बॉडी में क्या होता है, वी आर हैविंग मेमोरी सेल्स. मेमोरी सेल्स क्या करती है, मेमोरी सेल्स प्रोड्यूस साइटोटॉक्सिक टी सेल्स. सो साइटोटॉक्सिक टी सेल अटैक करता है किस पे, इन्फेक्टेड सेल्स पे. फिर क्या होता है, जो मेमोरी टी हेल्पर सेल्स है, दे स्टिमुलेट एंटीबॉडी स्क्रीशन. जब एंटीबॉडी स्क्रीट हो रही है यहाँ पे, सो देन एंटीबॉडी क्विकली न्यूट्रलाइज द इन्वेडर. मीन्स ऑर्गेनिज्म आया, मेमोरी सेल्स हमारी बॉडी में है, मेमोरी हेल्पर टी सेल, वो क्या एक्टिवेट करती है, फिर वो एक्टिवेट करती है साइटोटॉक्सिक टी सेल. फिर साइटोटॉक्सिक टी सेल के अंदर जाता है. यहाँ से फिर क्या हो जाता है, जो हमारी मेमोरी सेल्स है या जो हमारी साइटोटॉक्सिक टी सेल्स है, दे विल बी एबल टू देन प्रोड्यूस द एंड न्यूट्रलाइज द एंटीजन with the help of antibodies. So this was a normal process how things are happening in the nature, but we are having what now? We are having edible vaccines. So what edible vaccines do? When the organism is in the mucus line, the M cells the M cell passes antigen to the macrophage. जब M cells ने क्या लिया, जो M cells हमारी बॉडी में है, वो क्या करती है, वो इसको ऑर्गेनिज्म को लेती है, then it passes as an antigen to the macrophage and B cells. फिर जो ये macrophage है यहाँ पे, you can see here the macrophage cells now display piece of antigen to helper T cells. ये piece of antigen place करती है to the helper T cell. फिर हेल्पर टी सेल जो है, इट स्टिमुलेट बी सेल सीक आउट एंटीजन एट डिस्टिंक्ट साइट. देन एक्टिवेटेड बी सेल जो है हमारी, दे विल रिलीज एंटीबॉडीज एबल टू न्यूट्रलाइज द एंटीजन. सो सिंपली क्या होता है, जो हमारी एम सेल्स है वो एंटीजन देती है इधर बी सेल्स को, और यहाँ से वो एंटीजन देती है किसको, मैक्रोफाज को. मैक्रोफाज और जो बी सेल है वो क्या करते हैं, उसकी प्रोसेसिंग करते हैं. देन जो ये एंटीजन है, पीस ऑफ एंटीजन देते हैं किसको, हेल्पर टी सेल को. जब हेल्पर टी सेल को देते हैं, वो हेल्पर टी सेल क्या करती है, बी सेल को एक्टिवेट करती है ताकि आप एंटीबॉडीज़ बनाओ इसके तहत. फिर जब बी सेल्स एंटीबॉडीज़ बनाती है, फिर विद द हेल्प ऑफ दिस वी आर हैविंग एबल टू न्यूट्रलाइज द action of this antigen inside our body. So this is how our edible vaccine works in our body. This is having same mechanism we have studied. So Even it has been observed that some vaccines provide long time protection. Like in case if we have taken the vaccine for tuberculosis, we don't have to take again and again. It is lifelong protection. But there are some diseases as well for which we have to immunize periodically. Like cholera is there, tetanus is there. So हमें क्या, what we have to go there, we have to re-administer periodically. Means periodically हमें इसका लेना ही लेना है. Now we have to introduce gene in our body. We are having foreign protein in our plants हमें करना है. So we use two different approaches. हमने यहाँ पे use किया है. We have used two different approaches to insert or to express foreign protein in the body. So in first case, what we have used, we call this as in edible plant vaccine technology. Edible plant vaccine technology, and the second technology is called a chimeric virus particle, CPV technology. हमें क्या करना है, जीन को लेना, उसको इंट्रोड्यूस करना कहाँ, प्लांट सेल में. वी आर हैविंग टू डिफरेंट मैकेनिज्म्स. फर्स्ट इज द एडिबल प्लांट वैक्सीन टेक्नोलॉजी, सेकंड इज द chimeric virus plant technology. In case of जो हमारा है ये EPV, what we do, we take antigen from pathogenic organism. इसका DNA फ्रेगमेंट लेते हैं या स्ट्रक्चरल जीन लेते हैं, उसको फिर वेक्टर में डालते हैं. सिंपली वी टेक ए सबयूनिट वैक्सीन, एक्सेप्ट द न्यूक्लिक एसिड, वी प्लेस इट इनटू द वेक्टर. देन we mostly use, in case of plants, mostly preferable vector is the Agro bacterium mediated vector. So we have taken this Agro bacterium mediated vector into the potato plants, where this vector were able to integrate this transgene into the genome of the plant. So what we do, it can be expressed either in leaves or in tubers. Agar if the gene is expressed in the leaves, then we have to go for extraction of foreign antigen. Or if this transgene is expressed in tubers, feeding of edible tissue, तो हमें क्या feed करना है, अगर leaves में है तो उसको extract करना है. In both cases we were having immune response. These were able to generate the immune response. This is how edible plant vaccine technology works. Now the another example, we are having the chimeric virus particle, CVP technology. हम लोग कोई भी वायरस प्लांट लेते हैं, उस प्लांट वायरस को क्या करते हैं, हम लोग chimeric जीन में कन्वर्ट करते हैं. लाइक वी टेक एपिटोप, वी फ्यूज इट विद वायरल कोट प्रोटीन. फिर इसको हम लोग क्या करते हैं, इन्फेक्ट करते हैं प्लांट. मीन्स प्लांट को किससे इन्फेक्ट करते हैं, जो हमारा मॉडिफाइड ये है वायरस है. देन दिस मॉडिफाइड वायरस विल बी एबल टू एक्सप्रेस एंटीजेंस देयर. सो आइसोलेटिंग दीज chimeric virus particles then introduced into the humans will be able to initiate the immunological response. हम लोग ऑर्गेनिज्म का स्ट्रक्चर प्रोटीन या सब यूनिट प्रोटीन, या हम लोग chimeric viruses को यूज़ कर सकते हैं फॉर द expression system in the plants. We are having different examples of vaccines which are produced by the recombinant DNA technology in the plants. Like we are having first edible vaccine was Norwalk virus. So इस वायरस में क्या हुआ है, they have used genetically engineered potato where they are able to produce antigens or antibodies against the Norwalk virus capsid gene. इन्होंने इसमें यूज़ किया है. They have introduced this Norwalk virus capsid gene into the leaves or the tubers of the potato. So when the experiment were carried out of they have used 20 volunteers for this experiment. Out of 19, जिन्होंने क्या लिया, दे हैव एबल टू सी द एंटीबॉडीज़। 19 में इन्होंने देखा कि काफ़ी अच्छी क्वांटिटी में एंटीबॉडीज़ है। इवन आउट ऑफ 19 दे वर एबल, in 6 they were able to detect antibodies in their faces as well. So in their faces as well, so we can say this Norwalk virus capsid protein का जो है, it works for the immunological response, and we can use this capsid core protein for the initiation of, or we can use this for as a vaccination in the form of edible, means edible vaccine के तौर पे हम लोग इस प्रोटीन को यूज़ कर सकते हैं. Now the another example is our the hepatitis virus. The hepatitis is a single most important cause of viremia in humans. So if we will estimate it all over the world, we are having 300 million carriers from all over the world. We are able to produce hepatitis virus in tobacco. When we are having, like we are using tobacco as a system where we are inserting our transgene into it, due to this insertion, due to the production of hepatitis, due to the production of vaccine for the hepatitis in tobacco, they have reduced the cost. Even we can say penny per dose, it's nil. Plant system is, क्योंकि plant grow कर रहा है, वो vaccine भी बना रहा है अपने system के साथ. If we will be using this production of hepatitis B virus in case of fermentation technology by using microbes, there is those vaccines will be cost effective. Instrumentation cost, labor cost, हर एक चीज़ देखनी है. Even we have to be specific whether these fermenters are not attacked by the antigens. So by using these plant technology, we are have reduced the cost of the vaccinations. So scientists have made transgenic luteins which carry the hepatitis B antigen, and it has been observed they have shown the very good response. The another vaccine which are edible vaccine, we are having edible vaccine, it's for also cholera. So the causative agent of the cholera is the Vibrio cholerae. It has been estimated that 3 million infants die per year due to the cholera. So William and his co-workers inserted a gene into the potato in the tubers, which were able to form non-toxic component of the cholera toxin. Then these scientists used this as a edible vaccine. It can be said that like scientists have calculated, if we will have a cooked potato a week for a month, it should provide enough B cell activation against the cholera toxin. But we can, as we know, there are some vaccines we always need to get boosted up. So cholera is among them. So our immunity will fall over time. So for cholera, we have to go for periodic boost ups. So it's not lifetime like we have seen. In case of tuberculosis, we have used it. If we will have short ones, so we will be having lifetime protection. But for cholera, we have to go for periodic boosting. So we can say now the advantages. We can deliver to live or active vaccine into the human body, because these are not altered. We are directly taking our food, munching our food. So antigens are coming into our body, which directly initiates our immune system. These edible vaccines are cheaper than the injected type of vaccines, and it's easily available as they are heat stable as well. These food vaccines save millions of people who die because of lack of traditional vaccine inoculates. As a normal common man are not able to have costly vaccines, but due to this development of edible vaccines, or we can say this edible vaccine will be reached to the whole world, either developed countries or into the developing countries. So we will be saving 17 million people who dead every year due to the infectious diseases. So the day will come when we all will be having edible vaccines. Then the traditional vaccination process will be overcome by using these edible vaccines. Thank you.